First, I want to congratulate all the students who participate in this science fair. And I think you should be very honored to be part of this process. When I was at your age, I didn't have the opportunity to participate in any international competition, even domestic competition. So I didn't have the opportunity to go through this, which I think a very meaningful process because you will have the opportunity to think about the project and then conduct an experiment and then organize the data and then present the result to your peers. So that's a very important training for you to be a good scientist. And most importantly, it's the process for you to learn if you are really interested in science. And I think the reason we become interested in science is because you have the desire to solve some important problems, or you have the curiosity to learn something which you have no idea about why it's there or why the phenomenon is such. So I think it's a very good for you to go through this process to know yourself more about whether you should be in this area or not. Now, it doesn't matter if your interest is driven by curiosity or by the desire to solve major problems. I think the most important thing is that you have a strong interest and strong will to get into this field. And I want to take this opportunity to share with you my personal experience. I think this probably is the purpose I was invited to be here. I'm a chemist by training, and now become interested in biological problems. So we always think about how to use chemistry to deal with the biological process at the molecular level. And when you understand the detail of the biology, you become chemistry. And I think the fundamental training in chemistry is important for me to be able to attack important biological problems. So even today, I'm still in the field. And about 30 years ago, when I became independent as a scientist, at that time I was an assistant professor, decided to get into a new field, which was not popular. That is to do with carbohydrate chemistry and biology. And you know, in the biological world, there are three major biomolecules. One, of course, is nucleic acid, and the second one is protein. And the third one is carbohydrate. But nobody cared about the third one. So 30 years ago, I got into the field because I was curious about why nature has these class of molecules there and try to understand their role in the biological system. But there was no good tool. So today, you know, you have PCR, you have automatic synthesizer to deal with proteins and nucleic acid, but you don't have any good tool to deal with carbohydrate. So I thought I want to develop new tools so people can get into this field. And that's a major, major challenge because I couldn't make any story about why it's important. So when you write a proposal to the funding agency like NIH or NSF, I couldn't make a story. I never get funding. And every time when I wrote a proposal, I got rejected. But I was still curious why nature has these class of molecules there. So when you start as an independent career, you always had some startup fund from the university. So I used that startup fund to get into the field. And very fortunately, in the US, we had a system called Presidential Young Investigator. And that was designed for the young investigator who start first independent career. So I was very lucky to get that award. And I was one of the 7 recipients to receive the Presidential Young Investigator Award. And that gave me some confidence to continue what I was interested in. And I used the funding, and not only the funding from the government, there's also a matching fund from the industry. So I become familiar to DuPont, to Dow Chemical, and to IBM, and even to, at that time, GE Serol. So GE Serol, you probably remember neutral sweet. The sweetener, artificial sweetener, was invented by GE Serol. So I became a consultant for all these companies. And actually I didn't know anything, but I was invited as a consultant and participated in the consulting process to work with other consultants to deal with some specific problems. And I learned a great deal from that process. So I started to gain confidence and then continue my research. And when I become associate professor, I still didn't have an NIH grant. And that was very unusual. But I started to have an NSF grant. It's a small grant, but I had that. And then when I was promoted to full professor, I was very lucky that many funding agencies want to give me the money to do research. But at that time, I really didn't need much money, because when I need the money, I didn't get it. When I don't need the money, everybody want to give me the support. So I thought that's very funny, and that's something you may encounter in the future, because you are the cream of the cream. So I'm sure you are going to enter some unknown area, and nobody probably would care about it, and you have the insight to get into some very interesting area, and you don't have the peer to review your proposal. So you may have the same experience as I did before. Now today, what about carbohydrate? We know carbohydrate exists everywhere. If you look at our body, 90% of the proteins in our body have a sugar attached to that. And why is that? We still don't know. But why bacteria such as E. coli don't have this kind of so-called post-translational modification? And what is the secret about it? We still have no idea. And that is the reason I continue to develop new tools to understand the reason why nature has this kind of post-translational modification on human proteins. But today we know a lot of these type of molecules are associated with diseases. For example, bacterial viral infection, cancer metastasis, and differentiation or development. We know stem cells today. And cells from stem cell to mature cells, or from cancer stem cell to mature cancer cells, all have to do with this molecule. And so we start to be able to link these kinds of molecules to diseases. And because of that, the funding agencies become very interested in that, because you are going to understand the mechanism of some disease, particularly cancer, infectious diseases, and then use the understanding to come up with new strategies or new solution to the problem. So now you have you start from a curiosity-driven kind of approach to problem-solving kind of approach, and that's what we are today. And a lot of young scientists who get into the field, they move very quickly to the rank. So I'm very pleased that I chose the right thing to do when I was young. I had the courage to enter something which has a very high risk. We have no idea whether I will succeed or not. But I feel very fortunate that I was able to get into the new field and got some important contribution in this important branch of science. So I just want to share with you some personal experience, and I hope you have a very good time during these days, and good luck. Thank you very much.